Hi everyone, my name is Gabriel Gonçalves Dias. I'm a master's student from Brazil, and today I'll talk about my work entitled Targeting Spike Protein, an in silico approach towards the discovery of new anti-SARS-CoV-2 adsorption inhibitors. So, we all have experienced the spread of the SARS-CoV-2, and up to this moment, we've had more than 4.5 million deaths caused by COVID-19. Of the main strategies employed by the SARS-CoV-2 to establish the infection, the interaction between the spike protein and its human receptor AC2 is of ultimate importance, and thus blocking this interaction could lead to the prevention of virus attachment and infection. Unfortunately, we don't have an effective small molecule inhibitor just yet. Once experimental approaches are expensive and time-consuming, we use an integrated computational approach targeting the SARS-CoV-2 spike protein in order to find new drug candidates to treat and prevent COVID-19. For doing that, we first looked for SARS-CoV-2 spike structures from the protein data bank. For 135 PDB IDs, we got into only 21 structures, according to our inclusion criteria. Then we performed a confirmation analysis of these structures using the Bio3D OR package. Bio3D first calculates a conformational invariant core and then uses this core to superimpose these structures. After that, it performs a PCA analysis to obtain clusters of the main conformations. We did that for both chains from the trimeric spike structures and receptor binding domains of the structures containing the full and with no gaps or BDs. We used this strategy to select the main conformational representatives for a binding site prediction approach, using the default mode of the FTMap web server, followed by a site classification using the DrugPy plugin into druggable, druggable non-canonical small, borderline, and borderline non-canonical small. And then, we performed a virtual screening in 4 main steps. First, we chose to work with the Cambridge core and Express screening libraries. Then, we filtered the compound with a drug likeness filter, and after that, we used a docking-based virtual screening protocol with the GLIDE program using 3 different calculation modes. The high throughput screening mode, the standard precision mode, and extra precision mode. The best virtual hits were then selected for a ligand-based virtual screening using an in-house phenotypic QSAR model built with random forest and max descriptor. Finally, we've made an cluster centroid analysis and a visual inspection of the poses to select only a few compounds. We noted from the PCA analysis that the chains clusterized into 3 main conformational groups that you can see here. We observed, as we can see by interpolating a trajectory from the most similar structures in the PC1 space, that there is a major RBD motion in the spiked structures, as shown in the literature, followed by some N-terminal domain motion. Narrowing down the analysis to include only the structures containing completely modular RBDs, we observed 2 main clusters, as we can see here. We observed 2 main clusters related to an intradomain motion of a helix called gating helix, and described previously by Souza and co-workers to be important for the accommodation of a fatty acid. After doing the conformation analysis, we selected conformational representatives for a site prediction. We see here only the results for the RBD predictions, and we can note the 7 predicted sites, 5 in the structures containing an enclosed gating helix, and 2 exclusive to the structure with an open gating helix. 3 sites were druggable, and we focused on the most promising one, the fatty acid binding site, showing mesh in this figure. And in this figure. This site was then targeted in our docking-based virtual screening. So, at last, from 1.3 million compounds, we selected 19 virtual hits with the best properties, docking scores, and probability values that will be experimentally validated by our collaborators from North Carolina. So, in this work, we observed there are 3 main chain spike clusters related to the opening of the RBD and the closed and locked conformations of the spike structures. We noted too that there is an important intradomain motion of the gating helix of the RBD, which allows the exposition of a juggable site located on the previously described linoleic acid binding site. And finally, we conclude that by implementing an integrated computational approach, including docking-based and ligand-based virtual screening, we were able to select 19 promising virtual hits that will be tested experimentally. I'd like to thank CNPq, CAPES, FAPEG, and FAPESP for the financial support and all of you for the attention. Thank you.